Indeed. Let's now talk about aging with grace. Isn't that what we all want? We do that at Rejuvenate Biomed by developing medication that can have a big impact on age-related diseases. And we do that in a very specific way. I started this company because my father was not aging with grace, and I did not want that to happen to all of us. So in 2017, I established the company after 17 years at Johnson & Johnson, and this is where we are today. I have a wonderful team with experts in the field, so we have been able to push through this program really fast. On one side, we have our phase 2 asset, RJx-01, about which I will be talking a little bit more. It's today in phase 2 because of the wonderful data from the phase 1 study that I will be sharing with you, which is a little bit of special phase 1 study, so please bear with me. We also do have a preclinical portfolio, and they all have been developed based on our different platforms. We have 2 platforms, an AI platform and an in vitro platform, which I will talk about as well. What do we do different? Well, as you might have heard earlier during one of the keynote sessions, biology of aging has taught us a lot. We know that when we are aging, a lot of the damage control systems are not functioning optimally anymore. All these mechanisms are interlinked, and they are associated with age-related diseases. So it means that we will need to have an impact on multiple pathways simultaneously. So pleiotropic is definitely an important element. Since we will also not be able to find easily just one compound that can do that magic trick, we combine products. And we do that by starting with our AI platform. In the AI platform, and it also links back to one of the presentations earlier, it's not a black box. It's something that we have built. It's on curated data. We built new knowledge graphs with new knowledge included. And we have a very specific way of involving the interrogation of the system. The system helps us to identify compounds that can have an impact on multiple hallmarks simultaneously within a specific disease. In our in vitro platform in our lab, we then confirm that yes, indeed, we can have an impact on multiple hallmarks. If the IP is right, we move forward and we go into more traditional drug development. We do include phase 2, and then we look for a collaborator to do the phase 3. This way of working has led to our portfolio. So we do have RJx-01 in sarcopenia, and we also have other opportunities in other age-related diseases. Now let's today talk about sarcopenia. We do know sarcopenia from the aging perspective, the lower performance, the difficulty to get out of your chair, to open up a bottle, but there is something very similar, which is acute sarcopenia. It's the same thing, but it's accelerated. It gives us, from a scientific perspective, the opportunity to study this in a fast forward or in shorter clinical trials, but still can translate this to the chronic sarcopenia. In acute sarcopenia, you have multiple forms. You can have treatment-induced sarcopenia, and we will be talking about that later on in the panel on the GLP-1 side with the obesity. But you also can have disease-induced sarcopenia. For instance, people which have COPD, they cannot move around a lot, and they are chronic and acute sarcopenic because they get additional new hits because of the exacerbation and are not moving again. We always study cross-species, and today I will focus on humans, but definitely anybody that wants to know more about our C. elegans worms, about our killifish, about the different models that we use in mice, let me know. For us, it was important to be able to show cross-species that we have an impact on these mechanisms which are relevant for sarcopenia. More important, of course, we wanted to know, does this thing work? It's nice to know which mechanism it is using, but of course, it's equally important, even more important, to see if there's a clinical output. So also here, we studied this in multiple species. I will tell you a little bit more about the human trial we did, and then you can look at the data yourself and decide what you think about it. In this phase 1 study, since we are working with combination therapies, but the individual compounds have been around for quite a while, we do know the safety profiles. We also can look at the ADME tox to see if we can safely combine them. So safety is not such an issue for us. It's highly de-risked. So what we did is in healthy volunteers, I call them always brave because they gave us muscle biopsies, blood, did a lot of tests. And what they allowed us to do is to put their leg in a full leg cost for 2 weeks. We then have put them on compound or on placebo for 6 weeks, so during the immobilization and 4 weeks subsequently to it. After that, they go off and they get a personalized rehabilitation program. We looked at multiple factors here, primary and, of course, first time in humans, so we do look at safety, it's confirmed. We also have a new formulation. Because we know the products, we also knew how we would like the PK profile to look like. So these new formulations have been confirmed that they give us a PK profile that we were aiming for. Now, what everybody is most interested in is, of course, the exploratory data. In the exploratory data, we look both at the clinical outcome, but also at the mechanism. What we did here, and that's why I really call them brave, they gave us a muscle biopsy before the cast, in the casted leg. So before it goes in, it comes out of the cast, immediately a biopsy in the same leg, and then again 4 weeks later. If we now did an unbiased transcriptomics, we saw 3 things really popping up. We saw that during immobilization, as you would assume, inflammation goes up, fibrosis goes up, and mitochondrial health goes down. What we saw now with people that had been treated with RJx-01, it got back reversed. Having an unbiased transcriptomics telling us this is the best thing you can have. Of course, we were also interested, like what happens at the neuromuscular junction. So we did some deep dive there, and we saw that indeed the neuromuscular junction now was also being restored. So mechanistically, indeed impacting multiple mechanisms simultaneously, which were relevant. Now, other things we did is, of course, measurements on strength. So we asked the healthy volunteers to go into the Biodex system, and now we are looking at the movement or the regain of strength in the casted leg. So we asked them to push and pull against this lever. What we then can identify is pure strength. So, of course, after the cast, they have lost strength, and here we look at the regain of the strength. Again, you see that the gray line will always be placebo, orange will always be Rejuvenate RJx-01. So that's an easy way to recognize the colors later on as well. What we saw is you have spontaneous recovery. We know that. And you see it also very nicely in the placebo group. But you see that the RJx-01 treated group is doing better. Now, in our protocol, we predefined that if you would see an 0.1 value, this would be for us clinically interesting for this small group. Now, what is even easier to look at is a responder analysis. The responder analysis is on the screen. I don't think I need to explain to you what the conclusion is there. Then we did another test in the same device, but now we bring into account fatigability. So we ask the people, so the lever gets leashed, and now they have to push and pull their leg up and down 30 times in a row. We look at every moment in time, so the first movement, third, and so on until the 30th. The data that I show you here today are mainly focusing on the fatigued situation because that resembles real life. You look here at the rate of velocity development. That's the speed at which you can move your muscle. This is a very important one. We also look at our FD, our PD, because this means that if I would push you, are you able to put your leg in front of you fast enough? And then the other parameters are strong enough. What we saw here is that when people came out of the cast, there was already a difference between the two groups. That means that the compound was already actively activating pathways. We see that this becomes even more significant 2 weeks into recovery, at the end of the recovery, and then very surprisingly, as you might recall, during the rehabilitation, they are not on compound, but they continue to outperform their colleagues that were not taking RJx-01. These data, with many more data, of course, allow us to conclude that yes, RJx-01 does have an impact on strength, on performance, and on fatigability, which is exactly what we look at in sarcopenia. In sarcopenia, there is also the element of muscle mass. Muscle mass is very minimally important in sarcopenia because we do know that strength and mass are not correlated directly. There is an indirect correlation, but it's not that if one goes up, the other one goes down. We compare, like if you look at somebody who's like a bodybuilder, he definitely has the muscles. A weightlifter has the strength. Might have a different body though. but still that's how you can look at it. If we looked at total body, and we have to be clear here, so we have only been treating for 6 weeks, which is really short, you see that what you would expect in the immobilization, that in gray, the placebo goes down on lean mass. You will also see that fat mass goes up, and if you look at body composition, it goes down. RJx-01 now was bringing the opposite direction of these data. Again, a small group, small absolute numbers. These are non-obese people. These are just healthy volunteers. So we are curious, of course, to see what else comes up. But these data and many more, we of course already upfront have identified our IP. Our IP is secured, the grantings are in worldwide, and we have a protection here until the end of 2039. Of course, you can have a prolongation with the extra 5 years. We also have, on top of that core, we have an additional protection, which is the protection of the formulation. Now, important to know is that the 2 individual compounds, if we combine them, they are protected, but also the classes to which they belong are also protected. Now, with that being said, what are the opportunities for RJx-01? RJx-01 has multiple opportunities. The first one, for instance, in acute sarcopenia. Here we have been selected, so we're honored to have received that reward, to perform a study in COPD patients, a phase 2 study. It's fully non-dilutive funded, so we call it a gift from us to our investors, where we are looking at 80 patients which have COPD. Study is ongoing and the data will be available end this year. This is going to be a major moment in time for all of us, because now we can have it in a different setting. But there are other opportunities, and for the other opportunities, we're currently raising funds. So we would like to take a look and see if indeed we can protect the muscle in people that are taking the GLP-1s. We were mainly focusing here on the older population because you don't want to tip them into the sarcopenia. But there are broader opportunities. The disease-induced sarcopenia, there is something which is called osteosarcopenia. And we know that if you have osteoporosis, mortality goes down quite quickly. With sarcopenia, even more. And with osteosarcopenia, it's even worse. Osteosarcopenia is something that more women or females are suffering from than males. So we think that this is definitely a point of attention that we need to bring in. Next to that, of course, you have the normal, what we call age-related. So what do we want to do with the 30 million? As mentioned, we want to raise it as soon as possible. People always ask me when. I'm like, yeah, now, please. Because we are so ready to start those other clinical trials. We do have the material ready. We really want to move forward and open up the field further. So the upper part in orange is all about clinical things we can do. You see, this is very modular. With more money, we can do more. With less money, we can do less. But we definitely can spend the money wisely. We also will be focusing, of course, on our preclinical assets and bring them forward so that they can also go into the clinic. And our platforms are getting a lot of traction. We're one of the few AI platforms that have shown that what they predict also pans out in preclinical, but even in clinical. We know that we don't know everything. So we have a lot of people supporting us. We have key opinion leaders in the field from Sarcopenia, both on the US and the European side. But we also have drug developers, which are making sure that we make the right decisions, the ones that can look unbiased to all our data and help us move forward. As you can see here, we are not the super youngest management team, but do not worry about it. We do have young people, so definitely they will be working on our aging. So they're very enthusiastic, and if there's something I can say about my team, they are so motivated. They go for it. It's not only what they deliver, but also how they deliver it, and the science behind it, but also the way how we work on this ambition. This is for us more than just an idea that we work on. This is our passion. So takeaways for today, because I'm trying to get you back on your schedule, are 3 things. Why should you invest now? First of all, we have the data. Nobody has these data. Second of all, we have the team. This team has shown that it can do this. It has shown it also in the past. And it also has shown how resilient it is and how well it is able to pivot when a pivot is needed. The other part is, of course, well, there is one huge reason we are all aging. Even all these young people over here, we are all aging. So please, let us make sure that we are there for you on time so that you can age with grace. Thank you very much.